and the floor is yours.
Okay, thank you, Maria. I'm Lorenzo Veschini, and the work I will show you today has been performed in the past few years at King's College London in my lab. And it's very much concerned about modeling vascular microtissue in vitro, starting with a bottom-up approach, where we essentially build the vasculature, or what I will give me more detail on what I mean for which kind of vasculature, cell by cell, and eventually to get to perfusable microtissue, and that are very much similar to what we can observe in vivo, at least more biomimetic than what we are used to. And so without further ado, I will just go and perhaps share my screen first. That'll be a good idea. And here we go. Can you see my screen?
Yes.
And yes. And if I hover my pointer, can you see that?
Yep.
Yes.
Oh, good. So, okay. First of all, I will discuss for a minute why shall we be modeling the vascularized microtissue in vitro. So if we want to model tissue, human tissue somehow, both in silico and in vitro, well, because this is very useful for trying to understand better the disease or the biology of the tissue. And at the moment, the best thing that we have, or is becoming a little bit limited though, is animal models. So there, unfortunately, there is a little bit of incomplete overlap with human biology, and so certain questions cannot be really answered by animal models. Some, they tend to be expensive. I'm mostly thinking about mice, mammalian experiments, and so they tend to be impractical for screening approach, and they tend to be limiting their ability to investigate details of fast-paced dynamic phenomena. I'm thinking about molecular signaling, etc. And then there is some ethical concerns that are becoming more and more and more. And in this sense, to give context, just the FDA has recently announced that they are going to phase down a certain aspect of their drug discovery system approval, the animal experimentation, in favor of this new microphysiological system, which is essentially what I'm going to talk about to you today. And the promise of microphysiological system, or system that allow to capture some part of the biology of the tissue in vitro, in a dish, if you want, is that they are round-up approach, which allow very fine control of all variables. They are easy to manipulate experimentally. They can be used in screening. So if we want to give to our system many drugs, we can do experimentally. They are amenable to culture human tissue ex vivo. So we can, if we have the sample of human tissue, let me imagine cancer, these are amenable to, at least some of these are amenable to culture this tissue. They are inexpensive in comparison to in vivo studies. Again, in vivo, I'm really very much thinking about mice experimentation. So in developmental biology, I know that there are other kind of model system, but here I wrote in vivo, but I'm really talking about mice experiment, which is the one thing I have more in mind. And anyway, this system, the big promise for the future is that eventually we would like to create human organs in vivo, to even for transplantation. If you think about stem cells, we need to create bioreactors, if we want, that are able to kind of allow their assembly into more complex tissue and system. And at the moment, those that are available, I won't give a review of those, but if you can summarize, there is a limited ability to create truly biomimetic vasculature in vitro. There is a limited control of certain key experimental parameters like flow rate, and the initial cost to adopt certain microphysiological systems tend to be a little bit high. So not everybody wants to start, can start easily to do those. Okay, then a quick snapshot on why it's important to model the microvasculature. And then I will move to few experimental data that where we try to address this problem. So just to give context, I wanted to mention a clear difference between what we call macro circulation and micro circulation. That's why the macro circulation is what we tend to see in these kind of figures, where the whole big blood vessel that compose the human body, that are within the human body, that are essentially conduits for the pumping system from the heart to the whole tissue in the body. But the main function is transport to and from microcirculation. And there is no gas solute exchange and other activities, and they are specialized activities that tend to happen at the level of microcirculation. Just to go quick on this, here is a slide that normally I show because I want to highlight the complexity of the microvascular system. And in this upper panel, in this Bell et al., they made this very nice paper where they managed to image human embryos during development, and they were able to image neurons, endothelial cell, which compose all the blood vessel in the human body, and other structure. For example, here you can see these are staining of the blood vessels, and if you zoom in essentially in particular in this panel here, you can see how intricate is the network of blood vessel in the body. And in this rightmost panel, you see how intimate is the relationship between all blood vessels and their tissue during development in this case. So here we have the blood vessels, here we have the proximal epithelial bud, so the bronchi, or the precursor of the bronchi, and here the distal epithelial bud, which is the precursor of the alveoli of the lung, and this is a developing lung. And so if we want to model this in vitro, we have to keep this complexity in mind, okay? And the situation gets even worse if we want, because if we want to analyze at the level of transcriptomic, so the genes that are expressed at each time in the different cells, and in this case, it was done in a very nice work from Kaluchka and colleagues, where they took many endothelial cells from mouse, from very different organs, the brain and the kidney and others, and they performed single-cell transcriptomic, RNA sequencing, and they then try to figure out if they were different, the same, etc., and they find out that they are very different according to organs, and according to even within the same tissue. If you extract, I don't know, for example, the brain, there is a lot of heterogeneity going on, even the same tissue. And we are talking only about endothelial cells. So those cells that compose this tiny vessel here. And the structure will be evident in the next one, where this is a snapshot of how this is the neurovascular unit as a depiction. So how essentially the large circulation finishes and entering into the microcirculation that is essentially composed only of endothelial cells, which are depicted in red here, or in a little bit orange here. And these are omnipresent. And the pericyte, which are the green cells here, that are a little bit sparser. And in the case of the neurovascular units, there is the interaction with neurons by definition. But then if you go into other tissue, there will be the interaction with the parenchyma of other tissue. So here is to give a snapshot of how complex is the situation. And so what problem we might have. To address some of those, we started, well, we thought about some challenge that needed to be addressed. And some of those were to create a continuous network of pattern blood capillaries, which is feasible, and we optimize some system to do that. Reproduce in tissue-specific microenvironment, possibly. We just say that they are very different, the vessel in different parts of the body. Allow long-term perfusion and controllable of this tissue, microtissue, precise control of flow rates, allow possibly recirculation of immune cells, because the blood vessels are the conduits that also to bring bloods and all the immune cells to all the parts of the body, and to allow experimental perturbation as well. And from the logistic perspective, we would like our system to be inexpensive, possibly easy to implement, flexible, and function in a standard cell lab environment. So in other words, we try to make in such way that you don't need to buy a new lab to actually try to implement these technologies. And this is our prototype. And we developed this workflow, start leveraging on 3D printing. So which give a lot of flexibility on shapes and what is that we might want to build now or in the future. We decided about certain shape that we validate by computational flow dynamics, and I will talk a little bit more in a minute. And we produce 3D printed mold that then we use for soft lithography of polydimethyl siloxane, which is essentially rubber, and silicone rubber. And then we create a system that is depicted here, like in 3D, looks like a microscopic slide with something mounted in and some wells where we can operate and culture cells. And this is essentially, in one sense, similar to a standard cell culture. And this fits in a normal dish. And these are more technical details. And this is one of the most important parts, because this is the perfusion driver that allows us to perfuse with very controllable flow rates the perfusion of our system. And in this case, we use this micro pump, and this is controlled by a PID feedback loop controlled by a computer. And finally, you see that the whole system, beside the connector and the controller box that stays outside the incubator, it fits in a normal cell culture incubator. This is the validation in terms of what happens when the system is in theory exposed to certain flow rates. We did this by computational flow dynamic with very simple assumption of incompressible flow, and with a determined geometry. We started with our 3D design, and we assessed what was the predicted flow within the system. And we wanted to achieve certain flow rates that we tested, and tested certain dynamic range here. And we wanted to achieve a certain expected direct flow rates and shear flow rates in certain section of the system. And you can see this in C and E, that in different section of the system represent here. We have both direct flow, so orthogonal flow across the system, and flow tangential to the system, and we create shear stress in this case. Interstitial-like flow. And to move forward with the data that we got with this, a little bit more on the biology, that was all the validation and built up the system. We did use two cell culture systems to achieve the creation and perfusion of vascularized structure. One is the one on the left, which is based on fibrin hydrogel. And there is very extensive literature on this. There is a lot that is going on, and that is been published in several work. I would say that Roger comes from the MIT, is the precursor of this system, and they did a lot of validation. Is very nice system. There is one defect, or the one shortcoming, that is that the vascular structure built in this system, they tend to be, I would say, a little bit too big for being microvasculature proper. And this is because these systems are based actually in hydrogel, and they are very good at mimicking the first phases of wound healing, like skin wound healing, when there is a clot and a new blood vessel being formed inside that, but these are very disorganized. And this is so, it's very good to study this part of the phenomenon, but then they are very inefficient at studying the follow-up phenomenon, that is vascular maturation and build-up of more stabilized vascular network. So our part of our work was aimed at creating vascular structure in vitro that were more biomimetic. And in one sense, we remove some aspect of the previous system. So we get rid of fibrin hydrogels, and we create a whole cell system, so a matrix-free system, where we could co-culture different cell types, stromal and endothelial cells, to create very biomimetic vascular structure, like is shown in these images, and also here. And the last bit, let me see. Okay, this is the validation, so I can talk a little bit more on this. So we measure and study the vascular structure created in the two systems. And we, in fact, found out also by measuring that the capillaries, the vascular structure that are created in the system without matrices, it creates a structure that are more biomimetic in terms of the dimension of the capillaries. And also we could observe enlargement and maturation of the capillary structure over time upon continuous perfusion, which is a thing that hasn't, to the best of my knowledge, been previously shown in this system and in previous work. We had obviously continuously perfused also system in fibrin hydrogel, but we couldn't observe the maturation of the vascular network due to perfusive flow. And to conclude on this,
Can I ask you a question before you?
yes.
I guess just from my, so the fibrin hydrogel makes them larger. Does that mean it's making them mature faster, or they're just like bigger in size?
They are bigger in size, and
But it doesn't mean they matured faster.
No, it means that okay, because you see here from small they become big.
Right.
The problem is that the large vessel here are smaller than the small vessel here, so they are way bigger, and they are kind of enlarged sinusoidal structure, let's say, if we think about them.
Correct. I just was trying to figure out like what is the sort of like relationship between size, age, and maturity, because that's also important, correct?
Yes, probably I should define what I mean for vascular maturation, and I didn't.
No problem.
So at the beginning, when there is angiogenesis, and it happens normally in a very disordered way, there is a lot of vascular invasion in a newly formed tissue during development or during wound healing, which are kind of mimicking each other. And so one thing that happens that this complex vascular structure, it gets pruned over time. After it's created in a kind of excess to just make sure that everything works, but then it gets pruned over time, and many of the previously created structure disappears. And there are many mechanisms that are related to this, and I didn't show this slide today in the introduction, but one of these that is thought to be majorly important is perfusion. So once initially a blood capillary that is newly created is not perfused, but is perfused somehow soonish after it anastomoses with the neighboring vessels. So the perfusion creates biomechanical forces that instruct the cells to actually do something. And the one thing that they do, that they decide if to regress or to stay. And those that stay, perhaps they become slightly larger than they were at the beginning, while those that are regressed, they are simply lost and the cell possibly recycled. And so in this sense, I'm thinking about this moment in vascular maturation. There are also following up mechanisms that like recruitment of pericytes and starting the interaction with the parenchyma of the specific tissue. So in this sense, I was a little bit too generic in my maturation, and you did well to ask, because it needs to be precise, because it's a very kind of specific time frame, and it follows in principle what happens here. Experimentally, we jump this part to get into essentially what will happen in a later phase of wound healing, let's say. Make sense?
Yes, thank you.
Okay, and yes, more on this is we try to kind of find a matrix that will tell us, yes, okay, is the flow, might be the flow at least. We cannot measure very directly shear stresses, because people say, okay, are the shear stresses that instruct endothelial cells to stop doing vessel or to do more? And so we try to infer it kind of by in silico modeling. We created 3D meshes of our 3D structure in vitro, starting from images of our vessels. And then we subjected this this 3D structure to computational fluid dynamics in the exact same way we did for the validation of the chip here, but with a different geometry. Now, we know perfectly that the assumption that we take there, incompressible flow, non-elastic response anywhere, is not physiologic and is not completely telling the whole story. But the one thing that it says is that just by considering this simple model, just by having different geometries with different architecture of the network, we have very different perfusion of the network themselves. And this causes something that has been both predicted both mathematically by the group of Philip Maini and experimentally, I think, in vivo, that not all blood vessels are perfused at each time, especially when you are creating a new a new vascular network. And so more or less perfusion causes the maturation or even other phenomena during when the vasculature is actually mature, fully mature, let's say, and the architecture is somehow fixed or more stable. Not all vessels are perfused. And so this can create oscillation in the blood perfusion, which might be important biologically. And these images essentially show that the geometry of the network created in one system, the fibrin gel, and the matrix-free system are very different, and the latter recreates or gives back shear stresses and flow dynamics that is similar to what has been described in vivo. While this above, it seems that very low shear stresses and also the flows here are a little bit unexpected, let's say. And so this is just to wrap up. We had some requirement that we want to address, and we have managed to do something. We are still missing many things. For example, addition of stromal, other stromal cells, addition of pericytes, the perfusion and recirculation of immune cells is very limited at the moment. And anyway, we need to further validate the system, make it more robust experimentally. But we are kind of optimistic because we make this system quite inexpensive. So there is a way to improve and to test a new hypothesis. It's flexible. And so if we find out that we would like to do different geometries or a different architecture of the system, we can easily do that. And and it's easy to implement overall. And finally, I would like to thank all the people that have contributed. Many of these are kings, some of which are friends and collaborators somewhere else in the world, and some of which are just depicted in this photo. And thank you for your attention, and I'm really happy to take any question you might have.
Thank you so much. This was great. Are there any questions? I don't know how much of the modeling you're willing to talk about. I was just actually curious to know, more on a theoretical level, what would somebody do to distinguish what fibrin does to the vasculature versus the other? Like, are you working with physics? Is it like harder, softer? Like, I just meant, like, what are the attributes that somebody should think about in order to test such a model?
So I don't have an image that shows that, but let's stick to this. From experimentally, they are very different physicochemically, these two systems. One is an hydrogel that is very soft. Fibrin hydrogel used experimentally are very soft. They are seeded mixed with cells, and the cells spontaneously assemble into these vascular structures. So this is that's why it's a very good system and this is very robust, and it is also fast. It happens in 3 days. The point is that in this system, there are bits that are missing if we had to progress into the maturation phase, which is if we follow pathology or physiopathology, we know that the following stage is invasion of the clot, let's say, the fibrin gel, by stromal cell fibroblast, let's say. And this fibroblast will start to produce a different matrix. It's more collagen-based, and it becomes stiffer. So the initial softness, I think it's good for favoring migration of cells, and the endothelial cell are among the first, so they can create this immature vascular network. In vitro, we cannot, let's say, wait for this to happen or to make the passage, and this might be one way in the future, but I think it's experimentally inconvenient to just to start from the fibrin and allow it to remodel, because then experimentally we end up with other problems. And we start directly with the system where we can start mixing the endothelial cells with stromal cell that produce these other. And the other advantage I didn't mention at all, and I think is interesting, is that endothelial cells and the stromal cell fibroblast or mesenchymal stem cell, they can be obtained from different sources and different tissue, and we tried that. So we tried the experiments using different endothelial cells and different stromal cells, and it works more or less robustly with different cell types. And this gives us the sense that we can build more tissue-specific networks. In the sense, as we said before, different endothelial cells have different transcription state, and so they act differently. So in the perspective, in the future, to build a specialized vascular network, we would like to go into this direction. So these are my take over.
No, that's, I mean, I didn't know how soft they were, so that's that. So just again, for me, so the fibrin is very soft, and then the ova is more like, so you're able to control the stiffness, or it's just this other, it just somehow seems to be working to ensure that they're not growing so fast. They are creating these networks.
In one sense, they are very empirical measures, so very soft and
right. That's what I'm trying to understand.
very imprecise.
Okay.
I realize is very experimental and empirical. The fact that the difference with the OVA is that there are cells, and the cells is a multi-layer cell structure where the endothelial cell intercalate and produce their thing. So in this sense, it simulates development much more similar. No. And but again, I like to keep the parallel with wound healing, because there is a transition through this state in the case of wound healing. It's just experimentally we find out that it's more convenient to start some way later, and to achieve the goal that we had in mind, let's say.
No, that's the point I was missing. I think there's something cool about that. It's a cell infiltrating cells, and somehow that action seems to recapture what is happening in development. Sorry, yeah. I kind of missed that, but that is actually a nice point, which is different than what the other matrix is. You said it was a non-matrix, but I didn't clue in that it was a
that's why I call it microtissue. I probably should put a kind of I agree that is not clear. That is it's really a layered tissue, let's say 10. I imagine there are 10 layers of fibroblasts, one on top of each other. In the middle there are these tiny vascular structure.
And I think that this is interesting, because somehow you're able to recapitulate the size. So maybe the size is also an element of the space in between cells. I don't know. I just meant like maybe there are all these different elements that we didn't think about before. And somehow by having this more natural or some sort of a tissue or micro tissue seems to regenerate the size. Because for you, one of the things that you moved away from the fibrin is that size was an issue. And I was just thinking a lot about the size, but now I think it's even interesting just that having them to infiltrate the cells, maybe they're also limiting their size right away. I don't know, that you would probably know better than me, but
I guess so, in the sense that if I think development, everything starts small, and it becomes bigger due to different forces. Some are biomechanical, and as the heart becomes bigger, it starts pumping more blood, and this will help the different vessel in the body to become bigger. In one sense, at the beginning, they are always embedded within cells, even if in the embryo there might be, at the beginning in particular, there may be some ground matrix still. But yes, that was the point, to really try to simulate development a little bit more closely, and to enable to study them in a cell. So the good thing is if we can modulate the cell type and tune the speed, the flow speed, or the flow rate, then we can kind of, and we can measure them precisely. So in this sense is a very nice link if we have to kind of use this system to extrapolate data for in silico modeling. And it's where I think they become handy, because then you want to test a lot of cases.
I agree. Oh, Nadita, you have a question?
Nice talk. I have few questions. What is flown through these channels? Like these are vasculature, so it should mimic the blood. So does it have anything specific related to blood, or it's just a fluid like beer, PBS or something of that sort?
So you are,
Hello?
I understand well, you want to know what is the perfusion medium, what passes through the blood vessels.
Yes.
Yeah, is a cell culture medium, is the same that is, if we look, I know that is not the best representation. If we, let's see if I can zoom it. Yes, I can. So here we are, let's say this is a well you might have in your cell culture plate, and the cell goes in this little hole, and everything is surrounded by cell culture medium. And what we get, we perfuse with the same medium. So in terms, for example, of the perfusion, how it goes, everything is at balance, also by the fluid that stays in the well. So that's why there is this red box around, because the whole system is kind of flooded with medium on top, and this ensures that the kind of flow it's even, and it stays. It doesn't overflow, or it doesn't cause oscillation due to the pressure. Yeah. And which was the second
And
question? Sorry.
also, what is the exchange thing? Because vasculature is established to have the oxygen and nutrient exchange, but when you are providing a cell media which has already everything, so what will be the exchange kind of a thing which will be happening in this?
So you're asking what happens with the oxygen in the system, right?
Yes, or the
Yeah.
nutrients for that sake.
So that is a very, very interesting and well spot question, and is key somehow. We didn't address this question in this work, but the fast and simple answer is everything is cultured in normal oxygenation condition, which is the normal cell culture incubator, which we expect to have some 20-something percent of partial pressure of oxygen. And what we know is that even in oxygenated tissue, if we go really in physiology, at the capillary level, we should be around 10 to 7, something like that. Lung might be high, lung obviously is higher because it gets oxygen, but we are never at that level. Ideally, we can, and this is one of the development that will be really interesting to do, is to create put the system into a hypoxic box where we can lower the oxygen tension in the culture system medium, and at the same time put the reservoir of our fluid that we perfuse somewhere else in the incubator where the oxygen is normal, and then we will be able to mimic exactly as you say, the reality. And this, I'm sure, it will change the physiology of the system and will make it even more physiologic. In one sense, the fact that it's compact enough, the system that we're trying to develop is a good news, because then hypoxic box, they are not necessarily huge. There are some big ones, but it's easy to implement in a glow box style hypoxic box. if it answered your question.
Yeah, yes, I got it. And when you are culturing 2 or 3 cells, the patterning also changes, right? In the fibrin hydrogel, the pattern is based on the fibrin, the amount of fibrin and how it is patterned. But when you are going into a chip kind of a thing, the patterning, like how the pillars and other things are arranged, that also mimics how the cell shape and size is there. Are there any particular parameters or something of that sort where it is mimicking the real tissue kind of a thing?
I didn't quite get that.
Oh, okay. Let me repeat it. So,
Yes.
in a fibrin hydrogel kind of a thing, the way fibrin fibers align, the polymer aligns, is the way the cells get attached to the fibrin and then they grow,
Yes.
right? So, when you are mimicking into the organoid chip kind of a thing, are you changing the pattern or is it maintained in a specific state so that they regulate the size of the cells which are there?
In one sense, the system are very different. I agree with you that in the fibrin system that you have mixed fibers, and then the endothelial cell will attach and start nip and tuck the fibers to go where they want to go. And in the cell-cell system, there is a little bit of interlocking of cells, and then there is signaling from one to the other. To talk about alignment, we have noticed that for sure, fibroblasts or any kind of this kind of microtissue, they tend to form some form of biological shape. They tend to form streaks, and then this might be due to the particular cell culture substrate, but we don't explicitly try to pattern the substrate to achieve a special conformation of the system. And the matrix generate there, I assume to be for us random, but then let's say self-organized by the cellular system. What we observed that for sure, if you look at this system for a while, you tend to notice little kind of line of force happening. And actually, when we perfuse, we notice that the flow causes a little bit of movement in this. But these are more observation than actual data. But yes, this is also important, I think.
Okay, thank you.
Right.
Robin, you have a question?
Yeah. Hi, Lorenzo. Thanks for the talk. That was really interesting. This kind of follows on just from what you were saying. I was wondering, did you test if the fibroblasts, are they producing matrix components in the OVA system?
I'm not sure I did test it. No, well, I'm surely I've tested previously. I'm not sure that is in the paper. I don't remember that. The fibroblasts for sure produce some collagen, and we tested it, I think, by real-time PCR, and we compared them with mesenchymal stem cell at certain point.
Okay, and what's the ratio of the cells that you're seeding in these chips?
5:1, 10:1, depends from the cell culture system and depend from the endothelial cells. UVEC, they are a little bit more speedy. Other cell type, dermal microvascular or pulmonary microvascular, less fast. And so depend what is the aim of the experiment. You want to put less or more. And perhaps a different system you don't mind, let's say.
Yeah. And this, sorry, and just to cover, the OVA is just the fibroblasts and the endothelial cells. Do you have the pericytes in that model?
Not yet.
Not yet. Okay.
We were trying to create some, the one thing that we managed to do is to do that with whole stem cell system where we create endothelial cells and fibroblasts from stem cells. We put them together. It's not in the paper because it worked a couple of times and then there was need to finalize. And we were trying to do pericytes, so to have a whole same donor system, but it's for another day probably. But we didn't try yet.
Yeah, the more cells you try and pack in, the trickier it gets, right? So
yes, we try anyway. It works. The OVA independently from perfusion, it works well with pericyte. We did some mixing, but we didn't analyze it properly. So it's
Okay, great. Thank you very much.
welcome.
Thank you. Are there any other questions?
I have one, but maybe it's kind of naive. But I was just thinking of like in a regular wound, you might have chunks of more matured vasculature that are left behind. And in the current results you presented, it's all like de novo, right? So I wonder how chunks of mature vasculature might affect the network, if at all, and the fluid dynamics of it.
In one sense, is during wound healing, it will happen all with the fibrin intermediate. I was saying before, and there is invasion, right? So there is the wounded tissue that is none, let's say, and then it's kind of closed, put together by the cloth, and stuff comes from the sides. So this process is not reproduced by the model. It's all like, say, the cells have already arrived there. Let's say it's a matter of experimental convenience. And dependently on which kind of specific, there are people that are studying essentially how cells move and go and do these things. So if you want to study that, you might create a kind of thing to put the cells all, just to show the figure here. We tend to see everything with endothelial cell in between. The fluorescent things are endothelial cells, and they are seated in the chip and in between. But nobody prevents us to just put the fibroblast here instead of at day 5 putting some endothelial cell as well. And we will expect eventually that the endothelial cell from here will meet the fibroblast and create some kind of structure. It was one of the 2 ways that we wanted to do the things. We ended up landing on the one thing that gave us results faster because we needed to publish on it. But so there is a world when we can create a system that is good to investigate that other aspect. So it depends which time you want to investigate. I think is that's the good of the platform. I think one of the goods.
Okay, thanks.
Any other questions? I hope everybody saw how useful these systems can be. I'm already thinking about how they could be used to model, and you showed us a model. And of course, there's so many questions about what you can test and how you can maybe even use the model to help you improve the system, maybe. Yeah. So there are definitely a lot of overlap that is possible in here. And thank you for sharing. This was
Thank
great.
you. And I see it, this kind of new system. I focus on the vasculature, rather focus on, I don't know, pancreas, organoid. And actually, we try to put things together also in this context. And then it's good to have an in vitro system that you can measure easily, because then it becomes very easy to make transgenic things. They become fluorescent because you want to look at NFKB or whatever. And then you can measure it, the dynamic. And that is a thing that so we can measure the dynamic of certain signaling at single cell level. So this is the platform, but one of the platform, the possible platform. But then I think the systems are really useful for modelers.
I agree, and
I like the data that they want.
Exactly, I agree, and I really am happy, like, because I didn't catch it. I really like the idea that their cells, and these cells are infiltrating, and their size is readjusted. Like there's some, it's almost like a miniature organoid, but with some control, with measurements. I mean, there's accessibility in the system, so I hope others also see this.
Thank you.
Thank you.
I also wonder how much it scales, like the microtissue, how large can you get it?
There is one point that is one thing that I need to think next. Scalability was one of the first ideas, no? Ideally, it all started a few years back, but very, very few years back. But one of the things that gave me a good input that was a good idea to invest in this was a NASA challenge where they were asking for a 1 centimeter cube vascularized microtissue. And we are not there yet. I think this is 5 by 2 by few millimeters. So we don't get quite at the 1 cubic centimeter. And we need to find probably orbit and matrices that silicon rubber, PDMS, or we need to functionalize it better, and also better substrate than glass that is used in our system, because glass, yes, it works at the beginning, and then cell tend to lose their contact with glass, and then all that you created tend to kind of in sense. So scalability is an issue of, I think, material science in this case. So finding a good material that can be hold the cells, stick where they need to be at all times for the experimental time, and that will, and then this system should be scalable as much as you want, because then you can calculate the flow by CFD and create a system that scale up.
I realize that scalability is important, but I also don't want it to deter from its ability to right now capture these early stages that maybe haven't been captured before. I mean, I don't know. I mean, from what I gather, it's one of the first systems that you can see these smaller vessels or vasculature, and you can see them forming and growing, as opposed to just putting them on this fibrin and then they are just large. I just meant like maybe this little micro steps are also what do you call it? Sort of like keyholes you can actually peek through, and maybe you can get something from this early developmental stages that maybe we're missing in other steps. So I get the idea that it has to be larger and bigger, and we might want to get those other large big step, but maybe right now what you've done is opened up a time point or some sort of a developmental, steps that we haven't been able to access. And I think that's cool.
In another sense, we can think from cell biologist perspective, cell tissue biologist perspective. If you think about tissue, most of the tissue in human body are modular. So the liver is an assembly of liver functional units, the hepatic globule, pancreas the same, and so on and so forth, the skin as well. And so if you reproduce one of the functional units or few of them, then you have a micro world that represent the system in full, as far as you can multiply that. And that goes also along very well with modularity, with in silico, right, where you need to be modular in order to make things that are robust in themselves, and then you can multiply or call them at need. So it's
yeah, that's what I meant. Like sometimes maybe it's not about the size. It's right now what you've opened up a more, let's say it's I'm trying to figure like this time steps, like steps that we're able to potentially track dynamically that maybe we were skipping or missing. And all of a sudden we have a lot more time points and information for how these developmental changes are changing within this small window of time.
Better time resolution for a process.
Yeah, sorry, I wasn't saying this right, but that's what I meant. Like it's just all of a sudden you gave us access to these time points. And so there, I get there's always a trade-off, but there
is obviously where I show, and I should tell the story in a certain way, from all the few technologies that have been used for this, you can isolate sometime processes. To be honest, the OVA works also without perfusion. So you get the formation of the vessel. So if you are interested in the formation of the vessel, you can just do it in a plate, and you can do large screening on that. And in this sense, well, this first I have to mention, is in the paper, but we all stay on the shoulder of giant. I put together a few things, but the OVA is not our invention. We refined a previously existing system, and but it works without perfusion. So it's useful if you think that your gene of interest might affect endothelial cells. It's easy to do a transgenic and test it. All right, you have less angiogenesis, more angiogenesis, bigger, smaller vessel, and so on.
This is great. Thank you.
One point I was thinking about the modularity, what Lorenzo was mentioning about the liver and pancreas, this islets kind of a thing. is it possible to mimic such kind of a structures and study how these changes are captured, like in the early development as well, as the tissues form, the blood vessels are intricated. So that might be one of the cases where we can apply this kind of a system.
Is talking about the pancreas. I understood that that was the hint. Is
Yeah, but in the early development, when the pancreas starts developing, how this blood vessels and the islets are formed in the first place can be studied using this system.
we started
With
a
the,
project. Serendipitously, you asking me, we could have chosen any other tissue, but choose the one where we started the project that then is a little bit on hold right now, where we started applying this system, the OVA in particular, without perfusion yet, with and using it with IPC, stem cell derived organoids of pancreatic islet done by a collaborator at King's. They are specialized on that. We put them together and we wanted to say, okay, what is the angiocrine? So the vessel talking to the newly formed organoid and tissue and the angiogenic signaling and vice versa. And that is for sure a project in itself, very interesting to study. Which endothelial cell you need to put in? Is it possible to do a whole stem cell system and then drive? I want to ductal, I want to go beta islet because I want to transplant this patient and cure his diabetes. Yeah.
even the type 1 disease, type 1 diabetes has this kind of a change in the islet structures during the development as well. So that kind of aspect I was just thinking about.
Try to understand how it works, let's say. So the basic
Yeah.
mechanism of the, yes, that is. Let's say this is one model and it can be built on. That's what I'm saying. And for sure it's amenable to many applications. So that's why I hope it will be used.
Any other questions? Well, I want to say thank you, Lorenzo. This has been great.
Thank you for having me. It was very nice discussion and I hope you enjoyed.
Oh, I did. We did. I'm sure everybody did
Yeah,
too.
it's neat.
Thank you very much. So see you at the next occasion at the next seminar.
Yes, looking forward. Okay.
Bye-bye.
everyone.
Thank you. Bye.
Bye.